Good afternoon, everyone. Can you hear me? Semua boleh dengar saya tak? Kalau boleh, boleh thumbs up. Siapa yang tak dapat dengar boleh... okay, that's great. Thank you. Okay, we will start the session shortly. Thank you, everyone, for joining us today. Prof, are you okay to start?
Yes.
Okay.
Yeah.
Okay. Good afternoon, ladies and gentlemen. Welcome to the International Scientific Webinar organized by Persatuan Pegawai Sains Malaysia. I'm Anjali Ambaya. It is my pleasure to be the moderator for today's webinar entitled Introduction to Bioinformatics Application. Firstly, may I briefly introduce our guest speaker from Qatar University, Assistant Professor Dr. Rozaimi Razali. Dr. Rozaimi Razali is an Assistant Professor at the College of Health Sciences, Qatar University. Previously, he was a senior postdoc in the Medical and Population Genomics Lab at Sidra Medicine, Qatar. He received his PhD in Bioinformatics from Imperial College London, United Kingdom, from the lab of Professor Michael Stenberg, where he performed in silico studies of protein-protein interaction. Prior to joining Sidra, he was a postdoctoral researcher at the King Abdullah University of Science and Technology, Saudi Arabia, where he led multiple research projects, including the de novo assembly of tomato genome, as well as metagenomics and antimicrobial prospecting of the Red Sea. Prior to that, he was a bioinformatics manager at a startup biotechnology company, Cgenix, where he led the bioinformatics team, in charge of the analysis of next-generation sequencing that includes clinical exomes, whole genomes, and RNA sequencing. He had been involved in many peer-reviewed publications and is a participant in multiple internal and external research grants. Currently, he is leading key research with the population genomics and neurodevelopmental disorders research programs, in which he has been applying high-resolution data from short and long reads to enhance disease gene discovery and to build genomic references for the Qatari and local population. It is indeed a great honor that he is able to deliver a talk for today's session. Thank you very much, Prof, for accepting our invitation. Now I would like to invite Prof. Rozaimi to deliver his talk. Over to you, Prof.
Okay, thank you so much, Ms. Anjali. Let me share my screen. Can you see my screen now?
Yes, not yet, Prof. Just sharing.
Just 1 second. Okay, can you see my screen now?
I can. How about the others host?
Okay,
Yes,
I
now
think
we can. Yes, now we can.
All right, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. And I guess good afternoon, everyone. First of all, I would like to thank the Malaysian Association of Science Officers for inviting me to give this talk at this International Scientific Webinar 2023. It's really an honor to be giving this talk to all of you. My talk today is on the application of bioinformatics in providing insights into the historical migration and social development in the Middle East. So before we begin, I would like to briefly introduce myself. I know Ms. Anjali already did that. My name is Rozaimi Razali. I'm a bioinformatician by training. I've been in this field for 18 years now. I did my undergraduate at University Malaya, and I did my PhD at Imperial College. Majority of my bioinformatic career involved me working in several biotech startups such as InfoValley, GeneSec, Sangenix. So after around 10, 11 years working in the industry, I went and did my postdoc at King Abdullah University in Saudi Arabia. Afterward, I did my second postdoc at Sidra Medicine, and now I'm currently the assistant professor at QATAR University, where I'm involved in, other than teaching bioinformatic courses, I also do research involving population genetics, ancient DNA, genome assembly, and machine learning. Since I was, before we begin our talk, I was informed that the audience today are mixed. So I'm not sure how many of you are familiar with bioinformatics. So I thought of giving a brief introduction of bioinformatics. So if you were to ask 10 bioinformaticians the definition of bioinformatics, you will get 10 different answers. Okay, so this is my definition. So no two bioinformaticians will give the same definition. So this is my definition. You may or may not agree. For me, bioinformatics is a highly interdisciplinary field, okay, that develops and apply methods to understand and solve biological problems and questions. The word bioinformatics itself can be deconstructed, where the bio refers to biology and informatics refers to the processing of data to generate information. It combines multiple aspects of statistics, biology, computer science, mathematics, and other fields as well, such as archaeology, which is in my case, I'm interested in a combination of biology with archaeology. For me, the way I classify bioinformatics, I classify them into these three classes. Number one is what I consider as traditional bioinformatics. This is the development of algorithm and statistics, which assess relationship between members of large data set. The people who work in this subdomain, we call them as bioinformatician. Then the second class is those people who develop and implement tools by the first class, okay, in order to enable efficient access and management of different types of information. This subdomain, I call it bioinformation. And the people who work within this class, they are known by various different names. They are called bioinformatics system admin, bioinformatic programmer, bioinformatic analyst, bioinformatician, and so on. Then the last class is, or the last subdomain is computational biology, which involve the analysis and interpretation of various types of data, and the people who work within this subdomain, I categorize them as computational biologists. Okay, so now that we have gone over the definition of bioinformatics, let us see how bioinformatics is used, or how I use bioinformatics to provide insight into the historical migration and social development in the Middle East by studying the genomic history of QATAR. So at the moment, when we perform sampling, when we recruit patients, for example, for a genetic study, let's say we want to identify the genetic predisposition to type 1 diabetes, for example, we tend to group our sample into races and ethnicity, right? However, even between samples in the same ethnicity or the same race, you might have different variation, okay? You might even have two samples from different groups who are actually very much alike in terms of their genetics compared to sample within the same group. Now, this issue, we can resolve them by utilizing the concept of genetic ancestry. So let us look at the definition of race, ethnicity, and genetic ancestry. So race is a social construct which is based on some kind of physical trait. For example, here I'm showing skin color and it's self-reporting. Ethnicity is also a social construct and also it's self-reporting, but it is based on cultural trait. So for example, here you see all the Arab-speaking countries, okay? For example, people living in Morocco, in Tunisia, physically, they are different from people who live in UAE, Saudi Arabia, or Yemen, right? But they are all Arabs because culturally they speak Arab, right? So that is the difference between race and ethnicity. Now, genetic ancestry is more representative of the true nature of an individual because it involves a person's genetic makeup. Okay, so it consists of the genetic composition of an individual, which reflects their history, their historical migration and evolutionary process. So it's basically what they are born with. Okay, but the issue about using genetic ancestry is that you need to have high number of individuals in order to create this genetic ancestry. The reason why, because you need a statistical power to do this. Now, imagine if we can use a large genetic data set, okay, that we can use to group our patient or our sample of interest into genetic ancestry. Nowadays, there are many genome sequencing efforts being done all over the world. We have, for example, Genomic England, near to Malaysia, we have Singapore Genome Project, and in Qatar, we also have a similar project, which is called QATAR Genome Project. The aim of QATAR Genome Project is that by 2030, they aim to sequence all 300,000 Qatari nationals, right? So there is currently 2.5 million population in Qatar. 300,000 of them are locals, meaning Qatari Arabs. So their aim is to sequence all 300,000 Qatari Arabs by 2030. So in my when I analyzed their data set, I analyzed 6,000 whole genome data, and together with this 6,000 whole genome data, there are various features such as questionnaire, clinical information, imaging, and etc. So in my study, our aim was to understand the genetic structure of the Qatari Arabs, meaning Gulf Arabs. To achieve this aim, we constructed 5 objectives. The first one is to identify novel genetic variants that have not been seen in other world population previously. Second is to group Qatari nationals into specific genetic ancestry. The third is to study the run of homozygosity in the Qatari population. The fourth is to study the genetic admixture between Qatari genome and other genomes from around the world. So I mentioned 5, actually it's 4. Okay, so well, there are 5, but for this presentation, I'm just showing 4 out of 5. Okay, now let us look at how I solve the first objective, which is to identify novel genetic variants. So genetic variants, as I'm sure all of you are aware, they are differences in the genetic code, which makes each of us different. Our genome consists of 6 billion letters or 3 billion pairs of these letters. And these letters is composed of 4 letters, or we call it nucleotides. The random arrangement of this nucleotide define who we are. So why it is important to look for novel variants? The reason is because these novel variants could contain important answers with regards to drug interaction or susceptibility to certain diseases or susceptibility to certain pathogen. Okay, that is the reason why we are looking at novel variants. So here, as you can see in this plot, okay, as you can see in this plot, from our study of 6,000 Qatari whole genome, we identified almost 28% of the Qatari genome have novel variants, right? So around 25, 25, sorry, not 28. Sorry, 28%, or around 24.6 million SNPs are novel. So we have around 28% of the genome of Qatari samples are novel, meaning these novel variants are, what they mean is that they are different compared to other world population. In total, we identified over 80 million genetic variants from this 6,000 cohort. On average, you can see here, there is 20 disease-causing mutation that we detected in our cohort, which is slightly less than what was reported in UK 10K, because in UK 10K, they detected 25 disease-causing mutation on average. Most of the disease-causing are rare, with frequency less than 1%. Most of these disease-causing variants are autosomal recessive. Okay. Additionally, we also found 34 variants that can confer protection against diseases or symptoms such as malaria, obesity, and diabetes. Now, let us move on to the second objective, which is to group Qataris into a more specific genetic ancestry. So previously, there have been 2 attempts to basically group the Qatar genome into genetic ancestry. It started in 2010. Okay, in 2010, in 2010, they identified 3 Qatar ancestry: Qatar 1, Qatar 2, Qatar 3, red, blue, and green. Okay, but they were using 500,000 SNP markers from 168 samples. Right, this is SNP array. Then, after 6 years afterwards, in 2016, another study which uses 98 whole genome data so the difference between this study and the previous study, this study used whole genome data, but they still find the same genetic clusters or genetic ancestry. they identified only 3 genetic ancestry. Now, in our study of the 6,000 whole genome data, okay, what we found is that we found 6 possible genetic ancestry of Arabs. You can see the way that you read this figure or understand this figure, QGP is on the leftmost corner, right? So this is QGP, stands for QATAR Genome Project. This is our population of interest or population of study, and the rest are all the other world population. So all of this other world population came from 1,000 Genome Project. Okay, so you can see here, we identified 6 possible genetic ancestry. So you here you can see one is in gray, one is in blue, one is in red, one is in cyan. Then you have orange and a little purple. You can't see the purple here, but there is a very tiny line here. So basically, the way we group them is by looking at the fraction of the ancestry fraction. So if majority, for example, take one sample. If one sample has an ancestry which contains more than 50% blue, then they will be grouped under the blue ancestry. If someone has 51% of their genome is coming from the red ancestry, they will be grouped as red. Now, what if someone has 30% of blue, red, cyan, and the rest are orange and purple? So you don't have dominant ancestry. So for those sample, we group them under gray, meaning they are highly admixed individual. Okay, the problem with calling them blue, red, cyan is that it's very hard to compare with world population. For example, here, okay, all the greens are East Asian. All the yellow are South Asian population. All the purple are Europeans. Okay, so and all the, for example, under Africa, all the ancestry that are colored as yellow, as orange, they are coming from African ancestry. So for our QGP, we needed to identify, we can infer from here that the orange are the tiny part of orange here are coming from Africa. But what about blue, red, and cyan? They are not represented in the world population from 1,000 genome. Okay. So what we did was we overlay, okay, one way to do this is we overlay our population with other world population other than 1,000 genome. Okay. So when we overlay our population with other world population, we now can see there is an overlap with some parts of the world population. So the cyan, the cyan has been seen very in a small amount, okay, and they are predominantly in the Arabian Peninsula, in the Gulf, okay, in Jaziratul Arab. The blue, they are dominant in Arabia, in Northern Africa, okay, as well as in the Levant. Levant here, it means Palestine, Syria, Lebanon, Jordan, okay. Orange, as we seen previously, they are dominant in African continent. Red, they are dominant in the Levant, as well as in east of Euphrates. So in Iraq, there is two rivers, Tigris and Euphrates. So people who are from Iran, Iraq, Armenia, Azerbaijan, the Caucasus region, they are predominantly red. And then purple, they are dominant in the Levant and North Africa. So now we can identify, instead of calling them by colors, now we can identify or give them a name. So the Sayan, we call them as Par or Peninsular Arabs. Gar are General Arabs, sorry for the typo. The Web are West Eurasia or Persian, or we call it Ancient Khorasan. The orange are Africans, the yellow are South Asian, and the gray are ethnics. So from now onwards, I'll refer them by their respective names: Par, Gar, Web, African, SAS, and ethnics. Okay, so the analysis that I just did was looking at the autosome. I also look, because this is whole genome, I also look at the mitochondrial and the Y haplogroup. So when I look at the mitochondrial and Y haplogroup, they reveal distinct patterns of maternal and paternal ancestry. For example, let's look at the mitochondrial DNA. The major haplogroups were H, right, J, L, T, and U. Okay. All of this indicates maternal lineage, while for Y, it's predominantly J1. Okay, and I'm not showing you here, but the majority subclade is J1P58, which suggests a significant role for male in the ancient time. The male migration shaped the modern genetic landscape of the Middle East, while ancient Middle Eastern women, they remain relatively localized. Okay, so in ancient time, men are, and it makes sense because men move around, while women, they stay in the same locality that they were born in. Okay. Now, let's look in terms of the third objective, which is to study the run of homozygosity in the Qatari population. So what is a run of homozygosity? Run of homozygosity is a continuous homozygous segment in our DNA. So why it is important to look at run of homozygosity or ROH? The reason why it is important, because it can provide insight into the population demography over time. For example, if you are interested to look at the rate of inbreeding, as well as it can provide insights into recessive disorder. So when we were looking at the variant statistics earlier, we noticed that there are a majority of the disease causing were of autosomal recessive, right? So so that motivates us to study about run of homozygosity. So first, from the variant file, okay, from the genome, we call the variant. From the variant, we identify the inbreeding coefficient. We compare the QGP, the Qatar Genome Project ancestry, with all the other genetic ancestry. And we notice that the median, right? So the median for PA, the median for GA, and the median for WEB are very high, right? This is the inbreeding coefficient. Then we look at the run of homozygosity. So we divided our run of homozygosity into three: short ROH, medium ROH, and long ROH. So what does short ROH mean? Okay, short ROH means the ancient relationship to ancient people who went out of Africa, right? So the more short ROH an ancestry has, the more ancient they are, right? Because they contain the same haplotype that they inherited from the first people who migrated out of Africa. So at the same time, the further you are from Africa, the less in terms of ROH you will get. Okay. Then the medium ROH represent the background relatedness, and the long ROH represent rate of recent consanguineous relationship. Okay, so when we look at our ROH result, we notice that the PAR, remember, is represent the Arabs living in the peninsular Arabia. They have in terms of okay, so they have the median is very low, okay, which indicates that it contains the most abundant here, the most abundant ancient haplotype compared to even the modern African samples. So the YRI is the Nigerian, LWK Kenyan, even between the Kenyan and the Nigerian, and also QGP African, the PAR has a lower median in terms of short ROH. What this means is that they are more related to the ancient people who migrated out of Africa. Okay, so this means this PAR, the haplotype, is very ancient. Okay. Now we have looked in terms of the ROH. Oh, sorry. One more I forgot to mention. Okay, so that is for short. Now, if we look in terms of the long, okay, the long ROH, you can see the PAR, the GA, and the WEB. Especially the PAR and the GA, they have high rate of long ROH. Their median for long and their rate for long ROH is very high. Okay, so what this means is that it suggests recent consanguineous marriage have happened. Okay, but how recent and why? This is what we will try to answer by looking at the next objective. So the next objective is to study the genetic admixture between QATAR genomes and other genomes from around the world. So the genetic admixture is basically the genetic interaction between individuals. This can lead to emergence of new genetic variation and traits. So why do we study genetic admixture? So there are several reasons why we would be interested to investigate the genetic admixture of a population. In terms of the Middle Eastern or the Arab admixture, it is interesting because the first complete anatomical human remain was found in Ethiopia. Okay, it is located very close to the Arabian Peninsula. So here there is a very narrow cross called Bab al-Mandeb Strait. Okay, and yeah, so that's number one. Number two, Arabian Peninsula is where Mecca is located at, right? So for centuries, even before the birth of Prophet Muhammad sallallahu alaihi wasallam, Mecca has always been a site where people come for pilgrimage, right? So it's a well-known pilgrimage site even before the birth of Prophet Muhammad. The third reason is because the Arabian Peninsula or the entire Middle East region, they are involved in many international trade routes. For example, you have the spice trade route, then you have the incense trade route, then you have the silk road. Okay, these are all international trade routes that crosses through Arabia. And the last reason why we would like to study the genetic admixture of Arabs is because of the effects of colonization. So majority of Arab, Middle Eastern and Northern African countries, or we call MENA, one time or another, they were colonized by European powers. So there must be some kind of and they were colonized for hundreds of years. So there must be some kind of genetic admixture between the colonizer and the colonized people. So we ask the questions, how different are these genetic clusters, the QATAR genome project, the 6 genetic cluster from each other? Okay, that's number one. And number two, we ask the question, what is the genetic relationship between the QGP, genetic ancestry, with other world population, right? How much genetic contribution do this QGP ancestry provide to the world population, and how much or how many percent of the genomes of modern Arabs are influenced by other world population. So first we look at, we're going to look at two types of analysis. One where we try to identify QGP population as target, meaning how much foreign genome are inside the QGP ancestry. And then the second one, we use 1,000 genome as population. This is how much of Arabs' genomes influence other world population. So we look at the first one. When we compare the contribution of QGP genome into the world population, we notice that PAR, the Peninsula Arab, the Sains group, GA, the blue, and the Web, the red group, they played key role in shaping the genetic ancestry of the world population. Okay, so you can see here in terms of this is the so what you see on the top here, these are the target. Okay, so for example, for GA, the major contributor in terms of the genomic fraction is the Peninsular Arab. So this means the Ga and Pa are close to each other. In terms of the web is the West Eurasian Ancient Khorasan. They are also influenced by Pa, but predominantly they are influenced by the Tuscany ancestry as well as Northern European. For the QGP South Asian, they are mostly influenced. They are influenced also by Pa, Web, and Ga, but they are predominantly influenced by Bab, which is the Bengals. And for QGP African, they are influenced by Par, Gar, and Web, but predominantly they are influenced also by Yoruba in Nigeria and Kenyan. So what we can see from this analysis is that Pa is a major donor to Ga, which shows the close relationship between them. Pa is strongly involved in the admixture process of each of the QGP population. Okay, and interestingly, okay, when we try to look at which of the modern population contribute the most in terms of their genetics to Pa, we could not get any significant Z-score. What does that mean? This means that Pa, or the Peninsular Arab, they are the donor, they donate, but they don't, but very little of their genome is shaped by outsiders. Now we look at the, we use 1,000 genome and try to see how does Arab genome or the QATAR genome project ancestry contributes their genomic fraction to world population. So the world population that we did for all 25 population, but for the sake of time, I'm just showing you 4 examples. PJL is Punjab, LWK Kenyan, TSI means Tuscany, and PUR Puerto Rican. Okay. Okay, so what we can see here, okay, we notice that Pa, Ga, and Web, they play a key role in shaping the genetic ancestry of world population, right? Even the Puerto Ricans, you still have elements of Pa, Web, and Ga. The Tuscany in Italy, northern Italy, same, Pa, Web, and Ga. The Kenyan, Pa, Web, and Ga. The Punjab also have elements of Pa, Web, and Ga. So at least for these 3 Arab ancestry, Pa, Ga, and Web, they are involved in majority of the world population. The only one that doesn't have Ga and Web is the East Asian. But the East Asian, right, the Han Chinese, all of them, Japanese, all of them still have Pa. Okay. So why this is the case? So actually our result makes sense because many Middle Eastern and Northern African empires of ancient past, you know, many of these ancient empires were started in this region, right? The Assyrian, the Hittites, the ancient Egyptian, the Parthian, the Babylonian, the Eastern Roman Empire, Persian, and the Muslim empires. All of them started from the MENA region. Secondly, it also makes sense because Arabia, the Middle Eastern and Northern African region, they are, as we saw earlier, points of international trade route, the Silk Road, Sea Route, and Incense Trade Route. Right? We saw it here just now. Okay. So now next we try to answer how and when did they diverge from each other? Meaning we have these 6 QGP ancestry. How did they diverge? Why did they diverge? And when did they diverge? Okay. So to do this, we look at the effective population size. So from the variant file, we can basically trace the number of population across time. So effective population size, you can, I mean, in a easy layman term, you can think of it like the number of human, right? So basically, for example, here, the number of human during at 1 million years ago, based on the genetics of modern population. 1 million years ago, there were roughly around 20,000 human. All right. So the y-axis shows the number of population. The x-axis in the bottom reflects or represent the time. 1 million years ago, 100,000 years ago, 10,000 and 1,000 years ago. So you read from right to left. Okay, from right to left. Similarly, the x-axis on the top bottom, it represents the different ancient time period. So you have the lower Paleolithic, the middle Paleolithic, upper Paleolithic, Mesolithic, Neolithic, Chalcolithic, Bronze Age, and Iron Age. And here, the color that you have on top of the different time period is indication of the glacial climate. Okay. So you can see around 1 million years ago, population boom. Okay, and then something happened. Okay, which we see reduction of population starting from 300,000 years ago, and it reached rock bottom around 100,000 years ago. Now, if we look at the paleo climate during this time, what is happening? So two things are happening. Okay, so if we look at the sea level, okay, around so the sea level also you have to read from right to left. Okay, while the temperature you read it from left to right. Okay, so what is happening circa 100,000 years ago? So land bridge was opening. If you remember early on, we look at that there was this very tiny strait between Yemen and Ethiopia. Right now, it's sea. It's covered with water. But 100,000 years ago, you can walk, right? And that is how the ancient people moved from Ethiopia into Yemen. At the same time, the ice age was starting to form. The volume of ice increased. Okay. Okay. Now, around 7,000 years ago, we can the sea level was at its lowest. Okay. At the same time, the ice volume was at its peak. So so basically what is happening during the upper Paleolithic, population started to boom again due to favorable climate. So it's is a good time for hunter-gatherers because during Upper Paleolithic, humanity only practiced hunter-gathering. Okay, so there were lots of plentiful of vegetation, of animals that hunter-gatherers can hunt for. Okay, so because food was abundant, so population grew. Okay, but can you see around around 20,000 years ago, 20 to 6,000 years ago, population they stabilized sometime around the mid Mesolithic period. Okay, so during this time, two things is happening. So population stabilize, and but can you see the PA? They also basically never increase in terms of their population. So population of PA is lowest compared to the rest of the QGP ancestry. What is happening here? We hypothesize the reason why all other population the number increased while the PA got stagnant is because the rest became farmers, while the PA remain as hunter-gatherers. So let's look at again at the paleoclimate. So read from right to left. Around 130,000 years ago, this is the rain that the Arabian Peninsula are getting. Right, the Arabian Peninsula gets high amount of rain. Okay, um, around 20,000 years ago, there is a desertification, aridification event occur. Okay, so rain, basically rain is not happening. Okay, so this is the reason the aridification event push people either back to Africa or north to the Levant or to Yemen. Okay, uh, so and this it 20,000 years ago, it mirrors to what we have today. Okay, so we hypothesize that the PA they remain and become hunter gatherers, while the Ga, the Web, the SARS, the African they went back, the SARS they went to India, to Pakistan, Afghanistan, while the Web and Ga went north, okay, and started farming. So as a result, what happened to the PA? Because most of the population are left the Arabian Peninsula, they have to marry between each other, between the PA tribe. So this is the birth of the Bedouin people, right? This is how Bedouin came to existence, which makes sense. That explains the high rate of long ROH that we saw previously. Okay, that explains why the high rate of inbreeding. So we kind of answer how recent is the parental relatedness. It's around 20 to 6,000 years ago. So here is an image of the population split. Okay, so what we can see here, this is the point of view of different QGP ancestry. If we just look at PA, so when the ancient African went out of Africa, they split with the other African that stayed behind. So that basal population then split further, right? It became two. One went eastward to East Asia, and the rest state. Then around 50,000 years ago, we see a split between the South Asian ancestry with the rest. So the rest stayed in Arabia, the South Asian went to India, and around upper Paleolithic, when population boom, people start to spread all over the world. So you can see the split with Europeans, splits with the Iranians, and finally around the Mesolithic near to the Neolithic time, the split between Pa and Ga occurs. So what can we say? The Pa and Ga were closely related group. The ancestor of Pa and Ga splitted with Web after Web splitted with European. When some human migrated into Arabia from East Africa, they splitted into largely two main groups. One is the ancestor of East Asian, and then the ancestor of the rest here during this period 70,000 years ago. During the Upper Paleolithic, population started to boom, as I mentioned, because of favorable climate. So, as a conclusion, from this study, what we have showed is, despite the small size of Qatari national, the genetic diversity is huge, is very diverse, not huge, but very diverse, right? So this reflects the wider Middle Eastern region. So if we, for example, if a Qatari researcher wants to study the population living in Morocco, he doesn't need to go to Morocco or recruit Moroccan sample. He just needs to find Qatari nationals whose genetic ancestry mirror that of the Moroccan lineage. That's number one. Number two, this is the first genetic evidence for the early migration history of Arabs. Okay, I didn't cover the imputation, but we came out with an imputation reference panel for imputing Arab and Middle Eastern genotype. So in future works, we plan to improve the genetic ancestry prediction. We also plan to combine archaeology with paleoclimatology, with genetics and other data set, such as diet and disease, as well as improve our imputation reference panel. So yeah, so that's basically my presentation for today. So if there's any question, feel free to ask.
Thank you very much, Prof, for a very interesting talk. Just like it is a recap of the history as well as the ancestry and genetics part of the Qatari and also the world migration pattern. Thank you very much, Prof.
Thank you.
There are a few questions in the chat box. Before that, can I request the participants to turn on their videos? We are going to take a group picture as this is the last webinar series for this year. Thank you very much. We'll just give 1 minute for everyone to turn off the video. Yeah. Please turn on your video, please. Okay. Puan Masita, are you ready? Okay. We'll just give 30 more seconds for everyone to switch on the video. Okay. 20 more seconds. Okay. 1, 2, 3. Again, once more. Okay. 1, 2, 3. Okay. Thank you, everyone. Okay, let's move on to the Q&A session. Okay, Prof,
Okay.
I will address the question in the chat box, please, first.
Okay.
Okay, the first question is from TSME. How the development of bioinformatics application in Malaysia what is the most challenging part to develop it? As you have
Um,
experience. Yeah.
in Malaysia,
Yes.
in the context of Malaysia, the challenge is the fun, because bioinformatics requires, so it's not really the fun. It requires, yeah, it's actually the fun. To be honest, it's the fun. The fun because to do multi-omics analysis, for example, you need storage, you need processing power. Um, and this requires money to buy, right? So yeah, and the other thing would be the sharing of data. It's hard here to share genomic data, but I guess I think in Malaysia there's a lot of genetic data, but I guess it's not being shared. Sharing data is difficult. So, and these are the two main thing or main input that a bioinformatician like me require, right? Data and ability to process the data. Yeah.
Okay, thank you very much, Prof. And let's move on to the second question. Sorry, just missed it. Okay, as currently all are moving towards precision medicine, what is your insight about integration of multi-omics such as genomic, transcriptomic, proteomic, and metabolomic analysis in terms of processing a lot of raw data?
Yeah, I think this is an excellent question, because what I just showed you is just the first part of a three-part series. No, it's actually four-part. Um, so once we have done the genomic analysis using that ancestry, we and the same cohort, because the same cohort we got their genome, we did genome sequencing, we did RNA sequencing, and we did microbiome sequencing and proteomic sequencing. So we already analyzed the genomic. Now we are, me and my team, we are trying to publish a paper that looks at 3,000 of these 6,000, the transcriptome of these 3,000. So basically, we use the same ancestry and look at what are the differences in terms of the transcriptome. At the same time, there's another project where we plan to build reference genome for each of the ancestry. Okay, uh, and similarly, we try to look at the microbiome of each of these ancestry. What kind, is there any similarity in terms of the microbiome profile? So yeah, it's actually all are integrated, and I believe the integration of all these multi-omics data will provide a clearer, much better result in terms of precision medicine.
Okay, Prof. Okay, I hope that answered your question. And third question is from Mr. or Ms. Armani. Dr. Razali, again, how long does it take to complete or develop a QEP in whole genome population application? So she's asking basically how long does it take for the project completion?
So the project that I showed just now, it took me from 2018 until 2021. So 2018 to 2020, so 3 years to do the analysis, and then around 6 months to do the write up, and then another 6 months for paper review. So all in all, but I think analysis wise, around 2 to 3 years, maybe two. Yeah, if everything is, if you get everything ready, all the pipeline ready, it will take maybe like 2 or 2.5 years. That is my experience. Yeah.
Okay, do we have any more questions from the participant? You can unmute yourself and request your information from Dr. Rozaimi. Is there any more questions? Okay. Okay, one more last question from me. If a biologist like us would like to learn bioinformatics, which platform or courses would you recommend for newbies? especially for biologists, because we are not trained as a bioinformatician. So is there any website or a platform that we can actually learn how to do coding and so on?
Okay, so if you remember, when I presented one of the first screen or slide that I showed earlier, I classify bioinformatics into 3 classes, right? So first of all, you need to um, ask yourself, which of the class do you want to be? Do you want to be do you want to create software or algorithm, or do you want to create application, or do you want to use application to achieve or complete or analyze biological data? So there are 3 different bioinformatic classes. First, you need to identify where do you want to be. Okay, I assume based on my interaction and my experience, when most of you say you want to learn bioinformatics, that means you don't want to create code. You, you, you don't really want to create code if possible, or you don't want to develop a software. What you want instead is to use, for example, variant calling tool or RNA-seq tool to analyze your data. So it's more on the computational biology part. So if you are interested in that, become a master in computational biology, my advice would be to there are there are actually of um, a lot of tutorials you can use, and a lot of these tools are, you just need to master how to use Linux. Seriously, I based on my experience, I think if you know how to use Linux, you can run all this open source and free software, because many of the commercial software that we have today, that you have to pay money for, in terms of generating plots and everything, you can do it easily if you just learn Linux. I know, I'm sure some of you have heard about R, Python.
Yeah.
It really, for me, yes, if you want, you can, but there are tools that you can just execute without even need to learn Linux or R. Okay, sorry, not Linux, R or Python. Okay, you just need to learn Linux. And when most of the time when people say you have to use this R package or this R application or this Python application, what they actually mean is for you to execute those script. Executing the script is different from coding the script. Running R application or running Python application is different from coding. So you don't need to learn how to code. You just need to learn how to execute the code. All right. So so that would be my advice. For example, all the analysis that you saw just now, they were not done in R or Python, right? It's just using Linux. So because there are enough bioinformatic software, free open source software, that you can do genome analysis that I showed just now.
Okay. Okay, one more last question, Prof. Do you have the time? Okay. What
Yeah.
strategies do you use to ensure accuracy and reliability when analyzing biological data?
Okay, so for example, if I'm doing variant calling, okay, it depends on the depth of the sequencing that was performed. So if it is a research base, and we only look at data that was generated on average using minimum 30 x, for clinical 100x. Anything lower, if you identify a variant, you have to do Sanger sequencing to validate. Okay, but if the depth, the sequencing depth is high, you don't need to validate. Mm-mm.
Okay. There's one more question from Encik Mi.
Mhm.
Okay, is there any job vacancy at Qatar University? I'll leave it to Prof if he wants to reply. I think, sorry.
Okay. So usually, everyone can look at job the job portal at Qatar University. They just close, okay, because the recruitment is, so we have two terms in Qatar University. We have the fall period and the spring period. So our fall is usually the start of the academic year. So for example, if they want to recruit people for 2024-2025, they would start now in fall 2023-2024, so that by next spring 2024, they would already have a short list of candidate for 2024-2025. Okay, because during summer, everything is closed, because summer, it's hot here. For 3 months, nothing move. Everybody leave the Arabian region, um, including me. So nobody works during summer. So yeah. So I guess the next you should look at the job section of Qatar University or any university in Arabia. I'm talking not just Qatar, Saudi Arabia, UAE, Kuwait, Oman. You look during August to October. This is when they open. If you apply after these 3 months, you can apply, but nobody will look at it.
Okay. Thank you very much, Prof, for sharing the information. And ladies and gentlemen, thank you for participating in the seminar today. I hope you have gained insights into application of bioinformatics, and we are looking forward to your participation in future seminar. Today is the last session for year 2023. I would like to take this opportunity to congratulate the ISW committee for dedication and hard work in ensuring the success of this program. Thank you everyone for actively participating in the sessions held throughout this year. We are looking forward to meet all of you again next year. Have a good year and see you next year. Thank you very much, everyone.
Bye. Assalamualaikum.
Bye. Bye, Prof. Thank you very much, Prof.
Kaliptika dari